So last time I think we ended up right about here. Does this look right? That's what I wrote down anyway, that we were talking about gene expression, and we've got this chromosome that's depicted here on this slide, and the gene is right in this area right here, and that gene is going to be ultimately used to make some messenger RNA. And if that is actually a messenger RNA, then you know that the next thing you'll get is some protein that will be produced out of that messenger RNA. The messenger RNA is synthesized by an enzyme that's called RNA polymerase. And RNA polymerase basically synthesizes a single strand of RNA. It's a lot like the primase that we've already talked about. Primase does the same thing. It makes a single strand of RNA. In the case of primase, it's just getting that DNA replication process started. In this case, the RNA polymerase that does this reads the sequence in a very specific location. It has to find this region on the chromosome called the promoter, and the RNA polymerase binds very specifically to that promoter. Remember back to when we talked about enzymes and how enzymes have an active site? Remember that? And how the active site can only bind certain things. So RNA polymerase is designed to be able to bind this certain thing. There's a whole bunch of other DNA all along the chromosome, but RNA polymerase is designed to bind this specific sequence. So that's a group of A's, T's, G's, and C's that are arranged in just the right way so that RNA polymerase can sit down and bind to that and be able to begin its job. We can take enzymes and we can cut the DNA here, we can cut the DNA here, and then we can flip that sequence and make it point the opposite direction, and then RNA polymerase will come bind to it. And instead of going the normal direction, when RNA polymerase binds, it'll actually run off in this direction and start transcription in that direction instead of transcribing the gene that it's normally designed to transcribe. So it's just an arrangement of nucleotides, and if you flip them around, it tells RNA polymerase, hey, don't go that way, go the way that we're pointing now. And so that's an easy thing to do, very simple to do. But now RNA polymerase is going to combine here, and it's going to track along here, and as it tracks along here, it's designed to use one of the strands as a template and make a copy of RNA from that template strand. So RNA polymerase always synthesizes in the 5' to 3' direction. That means when it starts to make a messenger RNA, the 5' end is going to be the part that's synthesized first, and then it's going to move along toward the 3' end of that messenger RNA. You already know that RNA polymerases typically can initiate without a primer. That's why we get primase coming in when we're talking about DNA replication. It can start de novo and begin that process. But these RNA polymerases need that promoter site, this guy, to be able to find the genes in the chromosome and begin the transcription process. They stop at a sequence that's known as a terminator, and you can see the terminator right over here at the end of this region. And basically a terminator functions in a number of different ways. All of them kick RNA polymerase off of that strand of DNA. Some of them look a little bit like this, and you'll make this long strand of RNA, and then you might start putting in some nucleotides, and I'm just going to put C's in there because those are easy to draw. And then next you'll have something that can base pair, and so G's and C's base pair, and you start getting hydrogen bonds forming between these nucleotides, and you get what's called a hairpin forming in that messenger RNA. RNA polymerase is designed to hold on to a single strand of DNA. As this process is happening, the two strands bubble apart from each other so that they can read, the RNA polymerase can read the nucleotides that are down in between, and it's designed to hold on to a single strand of DNA and the strand of RNA that it's synthesizing. But as soon as this structure is formed right here, this hairpin, then the RNA polymerase can no longer hold on to the DNA and the RNA that's being synthesized, and so the system falls apart. RNA polymerase lets loose and it falls off the DNA, and then transcription stops. So that's one type of transcription terminator. There's another type of transcription terminator that looks more like this. A protein will come and it will bind to this terminator sequence. So again, a lot like enzymes that we talked about. Remember, enzymes have specific things, specific substrates that they can bind to. In this case, this protein has a site that specifically binds to that sequence of A's, T's, G's, and C's that we call a terminator sequence. it binds there, and then when RNA polymerase comes and tracks along and starts making a messenger RNA, RNA polymerase bumps into that protein, and it can't push that protein off because the protein is so tightly bound, and so RNA polymerase just falls off of the DNA strand, and again, that process terminates. That particular protein that binds right here is called Rho factor. And so this type of termination is called Rho-dependent termination because it requires that terminator sequence that Rho factor can bind to. The other type is called Rho-independent termination because Rho factor doesn't play a role in that. The protein doesn't have to come and bind. You get the RNA being produced, and then you get a hairpin that forms in the RNA, and that structure then makes the RNA polymerase unable to hold the RNA, and the things fall apart. So two different types of termination can happen depending on the specific terminator sequence that you're looking at. So RNA sequence is complementary and anti-parallel to the DNA template strand, and the DNA template strand is called the minus strand. This is just something you're going to have to memorize. The DNA template strand is the minus strand, and that's just the terminology that we use to compare them. And then the complement strand is called the plus strand, and the plus strand is going to have the complementary sequence to the strand that's going to be used for the transcription process. So when we say DNA template, that's the strand that RNA polymerase is reading to make the messenger RNA. Okay? And so the RNA then that's made is going to have the same sequence as the plus strand because it's going to be complementary to that minus strand. The only difference is that the RNA that's produced, you already told me last time, has uracil instead of thymine incorporated in, and it's also got ribose instead of deoxyribose. So in eukaryotes, in cells that you're familiar with, usually one gene ends up producing one transcript has one gene on it. So you might make a long transcript, and your transcript might look something like this, and it might start here, and over here you've got the ribosomal binding site. And so the ribosomes are going to come and bind on there, and they're going to track along the messenger RNA this way. And they're going to track along until they find in that sequence the first AUG. Right? That's the start codon for translation. And they're going to track along, and then once they start tracking along, they're going to make a messenger RNA until they come to a stop codon. And I'm just going to put S there for stop codon right now. When that happens, then that's the end of the protein coding sequence. The ribosomes are finished, and then that polypeptide is made from that process, and that's a typical RNA. Okay? So there's a few other things I'll tell you when we get a little bit further along, but in prokaryotes, many times we see an RNA being considerably longer, and it will still have the ribosomal binding site, and it will have an AUG shortly thereafter, and it will have a stop codon. But then on many RNAs in prokaryotes, you're going to very soon see another AUG pretty close to that stop codon. And if you go on down, you'll see another stop codon, and you might even see a third AUG and another stop codon along the way. And this type of messenger RNA actually codes for 3 different proteins. If you think about it, when you took the cells that you put into, let's use the lactose media from lab last week. You took cells out of a tube, you put them into the lactose media, you said use lactose if you can, and most bacteria aren't normally geared up to use lactose because lactose isn't around in the environment very much as free lactose. And so they'll usually use glucose first, you've already heard that 100 times, and then they're going to use lactose. But to do that, they have to turn on the genes that code for the enzymes to be able to break down lactose. And in reality, the RNA that I just drew here is a lot like the RNA that's produced when you feed the organism lactose as the only carbohydrate. they start making that RNA, and that RNA codes for all the genes. There's gene 1, there's gene 2, and there's gene 3, all on the same messenger RNA. So now as the ribosome comes and binds here and starts tracking along, it synthesizes enzyme 1, and then very shortly after it synthesizes enzyme 2, and very shortly after that it synthesizes enzyme 3, and the ribosomes don't actually fall off. They just keep moving along until they get down to the end of the messenger RNA to begin. So that in that way, prokaryotes, when they make this type of message that you've probably not seen before, they can turn on all of the gene products that they need to make for a process all at one time. In our cells, if we had a similar process that required 2, 3, or 4, 5, or 10 different proteins to be kicked on at the same time, we've got humongous chromosomes, and we've got lots of chromosomes. And so one of the genes, this one might be out on chromosome number 3, and another one might be over on chromosome number 5, and another one over on chromosome number 7, and they all have to be kicked on at the same time. So there's different systems to be able to do that. But bacteria is smaller, everything's more streamlined. They make one messenger RNA, and then they can make all those proteins that they need all at once. So that type of message is referred to as polycistronic. Polycistronic message is going to have multiple genes on it, just like the one that I depicted there. Okay? So, but in eukaryotes, most of the message is actually monocistronic, meaning it only has one gene on it. We'll show you some variations down the road as we get a little bit further into this. So, to kick on transcription for some things, you need special factors, and those factors oftentimes are given the name sigma factor. Sigma factor is basically a protein that recognizes a specific promoter sequence, and the sigma factor works to recruit the RNA polymerase over to that promoter sequence. So I don't know if it happened at your school, but we send out a lot of people to go to different campuses to talk to students and say, hey, our nursing program is awesome. You want to come to UMHB because we've got the best nursing program or biology program or business program or whatever it might be. We send people out. They act a lot like sigma factor, and you're the RNA polymerase, and UMHB in this case is the gene. They try to recruit you over to the promoter sequence of UMHB to get you here so you can do your job and you can be productive here at UMHB. So there are other sigma factors. The UT has sigma factors, recruiters. A&M has sigma factors, recruiters. They're more like constitutive genes. They're on all the time. They don't have to do as much recruiting because the polymerase normally goes there all the time. But some of those other genes that aren't normally as active are going to have sigma factors. And so that's an analogy that hopefully will help you understand. So sigma factor recruits the polymerase to come and say, use this promoter. So there's some examples of when sigma factors play a role. One, for example, is the process of sporogenesis. Bacteria that can make endospores don't always make endospores. They only make endospores when they're beginning to starve or the conditions are getting challenging. And so they don't want to produce those proteins that are needed to make endospores all the time. And so when it's time to make endospores, they'll make a very specific sigma factor that will now recruit the RNA polymerase over to the genes that are responsible for that sporulation process. Sigma factor loosely associates with RNA polymerase, and shortly after RNA polymerase starts the transcription process, sigma factor disassociates and it goes and recruits another RNA polymerase. So more of that messenger RNA can be produced. There are different types of sigma factors that recognize different promoters. I just told you about one of them that's involved in the sporogenesis process, but there are many other sigma factors that play a role in regulating other genes as well. So once they get started, then they control the transcription of a certain set of genes. In eukaryotes, we have something that functions kind of like sigma factor. There are proteins, again, that bind to some portion. These guys don't usually bind directly to the promoter, but they bind to some other sequence near the promoter, or sometimes actually a long way from the promoter, and recruit RNA polymerase in, and those guys are called transcription factors. They're specific proteins that can bind to different parts of the DNA around promoters and recruit the RNA polymerase and say, hey, come transcribe this gene now because we need this. Other genes we've already talked about are constitutive. Those guys don't need RNA polymerase to have a sigma factor to recruit it. And the reason they don't is the G's, A's, T's, and C's down in that promoter sequence are rearranged differently so that they naturally attract RNA and it binds more tightly and it finds it more easily. And so there's differences between promoters, and that makes sense because you already know that the cells want to use glucose when it's available, and it usually is. They don't use lactose unless there's nothing else available, so that, again, they don't need to turn on lactose very much. We'll talk about how that works down the road. So here's a scenario of transcription. We've got this chromosome that's double-stranded. One thing that they don't show you is that RNA polymerase, as it's moving along, the strands actually kind of separate and bubble out from each other because it's got to be able to read those bases that are down in between the two strands to be able to know what sequence to put in that message that it's synthesizing. But as RNA polymerase moves further down, the message obviously gets longer. Finally, it hits that terminator sequence that we've talked about, and depending on whether it's row-dependent or row-independent, either way it falls off, and the transcription process terminates. So I already told you that the promoter sequences are sequence-dependent, and you could flip them around again. We could take this guy and we could cut the DNA here, flip it around, and then instead of transcribing this gene, it would actually transcribe in the opposite direction. And we could do that for both of those promoters, and they're enzymes that can easily be used to do that process. In fact, we can just synthesize a promoter in the laboratory. We can tell a machine, make a piece of DNA that has this sequence that we know is the sequence of a promoter. It'll synthesize it. We can glue it back into another molecule, and then RNA polymerase will use that piece of DNA to make a specific protein. What they're trying to show, yeah?
So no matter what, even if you change the directions, it's always 5 to 3.
Yeah, so if you flip it around, it's reading the other strand as the template. You got it. Because of that nature of RNA polymerase. You got it. So in this case, that's a great question, Wade. Over here on this template, the way they've shown it, this is the 5' end, and this is the 3' end down here. On this RNA, the 5' end is here, the other is here, and so they've labeled the strands. In the case of promoter 1, the red strand is being used as the template strand. That means that's the strand that RNA polymerase is reading, and it's making a sequence that's complementary to the blue strand in this case. Everybody good with that? But on the other promoter, on the other side, the blue strand is being used as a template, and the RNA that's produced is going to have the analogous complementary, or the same sequence as the red strand on the top. The RNA is going to be complementary to the template sequence, and it'll have the same as the top sequence, except for uracil being substituted. Okay. Once the RNA polymerase hits that terminator, again, the whole complex dissociates and falls apart. So in that way, we build messenger RNA, and then the messenger RNA has to be read, and you know that process from anatomy and physiology is called translation. Basically, the messenger RNA is a temporary copy of the genetic information that's back in the chromosome, and it's used to do translation. To do translation, we need messenger RNA, we need ribosomes, we need tRNA, and we need a few accessory proteins that come in and help with this process. And some of those things that are described here in this table, and you can read through those, but we'll talk about those just a little bit. Before we start looking at translation more closely, let's look at the genetic code just a little bit. You should know from other classes that the genetic code is read with basically 3 nucleotides, and those 3 nucleotides that are read together we call a codon. We call it a codon because it codes for an amino acid, and that's how it got that name, codon. And the other thing that you could figure out pretty quickly is that if you take a 3-position region of DNA, and any one of those 3 positions can have 4 possible nucleotides put in, then that actually gives you the number of 64 possibilities. You can do the math on paper, and you'll figure out that there are 64 possibilities when you're looking at 3 positions, and each one of those can have independently 4 different combinations. And so if you look at it, that's what you're seeing in this table right here. If we look down this way, we see that in each of the second positions that is listed here, the second position is a U. But let's look at this first. If we look at this, everything that's in this panel that I just circled begins with a U. All the way across, all the way to over here, they all begin with a U. And if we look at the second panel of this, then everything that's in this panel, all the way across, here, 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 and here, all begin with a C. And likewise on down, the next one begins with A, the next one after that begins with G, and so on. And so you can count them, and there are a total of 64 possibilities. And then we get over here to this part right here, and we see that 3 of those code for stop codons. And this is E. coli, and a typical E. coli has 3 stop codons. And whenever the ribosome hits those codons, it stops the synthesis process. That polypeptide ends, and that's the signal to complete that polypeptide synthesis. I just want to check background information. Why does it stop there? What do you know? Why doesn't it just keep making the polypeptide longer and longer and longer?
Does it just not have a
An amino acid?
Yeah.
So what carries the amino acids in to the ribosome? tRNAs. And in E. coli, they don't have, typical E. coli doesn't have tRNAs that have the anticodons that match those codons. So because there's no tRNA, there's no possibility to carry in an amino acid. And since there's no tRNA that can come in, the ribosome sits there for a little bit. No tRNA comes in to bring the next amino acid in. And so the ribosome basically falls apart and releases that polypeptide. So I said typical E. coli has 3 stop codons, but I want to tell you a little story of how they figured this out. People were looking at viruses that infect E. coli. And some of those viruses could infect one strain of E. coli, but they couldn't infect another strain of E. coli. So they started looking down and trying to figure out what's the difference between these viruses. Why can it grow in this type of E. coli, but it can't grow in this second type of E. coli? And they started looking at the genome of the virus, and they figured out, well, this virus has picked up a sequence in the middle of its gene that's necessary for growth that's a stop codon. And so when we put it into this cell, it can't make that protein that's required for the virus particle, and so it can't grow in that cell. But then when they put it into the second E. coli strain, it grows fine. And what they figured out was that one of the other tRNAs actually picked up a mutation in that anticodon region, and now that tRNA could read one of these stop codons and put an amino acid in there. And when it put an amino acid in there, then the protein could be completed and the protein was functional. And so they figured out that there wasn't a stop in that one because it did have a tRNA that could fulfill the process of bringing an amino acid in. And so that's why stop codons stop the process of translation, is because there is no tRNA that is able to bring in an amino acid there. And so the stop codons, what they're showing you there are E. coli stop codons, and it depends on the organism that you're looking at. And this is the typical genetic code for E. coli. The next line is interesting. That says that there's redundancy in the genetic code. You should have already figured that out, because now we know there's actually 61 codons that code for those 20 essential amino acids. There's a total of a possible 64, but only 61 of them actually code for amino acids. And you know that there's 20 amino acids, so we can do some quick math. And that says on average there are 3 codons for every amino acid that is put into a protein. Everybody good with that math? 3.3333334 or something like that number. So that's redundancy. That's what redundancy is, that there's duplication. And if we start looking a little bit closer, we see something pretty interesting. We actually see groups of codons, and all 4 of these codons code for serine. And if we look a little bit closer, all 4 of these codons code for threonine. And if we go on over this way, these 4 code for valine, and these 4 code for leucine, and over here these 4 code for arginine, plus there's a couple more here that code for arginine, and these 4 code for glycine, and so on down the list. There's lots of redundancy there. And if we just zoom in and look at one of those, let's look at these 4 for arginine right here. What we've got there, the first one is CGU, the next one is CGC, the next one is CGA, and the next one is CGG. And what you see is that for arginine, as long as the first nucleotide of the codon is a C and the second one is a G, the third one can be anything. I'm going to call it X. The third one can be any 4 of the nucleotides, and the ribosome will still put an arginine in that position in the messenger RNA. And if you look over here for these 4 for leucine, you've got the same thing. There's CUX over here, and over here for valine, GUX. My handwriting's getting sloppy. GUX. And so in each of those cases that I first circled, the third position can be anything, can be any of the 4 possible nucleotides. And that's called third position wobble. And the hypothesis is that mutations happen. We already talked about mutations happening, right? Remember, DNA polymerase, it's error prone, it inserts mutations when it's making mistakes. And if you have this redundancy built into the system, at least 30% of the mistakes don't impact the protein that would be produced. Because if the mutation ends up in this third position, it's going to change it to another nucleotide, and you're still going to get the same amino acid being produced in that particular protein, or put into that protein. And for each one of those that I originally circled, the same thing is true. The third position can be any nucleotide, so a mutation changing the gene will not impact the structure of the protein. And so redundancy presumably helps the cell be able to survive through mutation processes in that way. All right, so we've got this RNA, and I kind of sketched this for you a little bit ago. We have a ribosomal binding site that's right here. It's going to come to this start codon, which is going to be an AUG in the RNA, and it's going to transcribe along until it hits this stop codon. And again, since there's 3, I'm just going to write an S there, and it stops, and the ribosome falls off and it releases that polypeptide. The polypeptide is folding as it's being translated, but there's sometimes some other proteins that come in that are called chaperones to do that. But let's look a little bit closer at what's happening when translation is going on. So we've got messenger RNA here, and somewhere out on this side, on the left, there is an AUG, a start codon that feeds into this. Everybody good with that? And so we're going to assume that that's true for all of these, and there's a start codon that feeds in. And basically, if you look at it that way, and this is the normal messenger RNA that's up here in this top panel, it's going to read along, and the ribosome is going to start translating at AUG, and then whatever codon is next, and whatever codon is next. And then when we get to this region, we get to this codon, and the first codon, CUG, codes for leucine. The next codon, GCA, codes for alanine, and so on down the list. So we've got leucine, alanine, leucine, proline, and threonine down that particular polypeptide that's going to be synthesized. But then when we go to the next part, if we assume that something has happened and we get a 1 nucleotide deletion somewhere here in the DNA, which results in a 1 nucleotide deletion in the RNA, then that shifts the reading frame. And what it does is it shifts the reading frame now so that we're getting these 3 nucleotides read as 1 codon. And I overlap, but these 3 nucleotides read as 1 codon, and these 3 nucleotides, and you can see those codons represented right here along the way. And what that does is it makes a completely different protein be synthesized out of that same messenger RNA. The only difference is a 1 nucleotide difference in that messenger RNA that may be thousands of nucleotides in length. And it shifted the reading frame because it put the codons in a different relationship to the AUG that's out in front. and so a different reading frame is read. And the same thing would happen if we get a 2 nucleotide deletion out here. That would shift the reading frame over 2 spaces, and now we would get this codon used, this GGC, and that codes for glycine, and the next one is AUU, which is isoleucine. The next one is GCC, which is alanine, and so on down. And again, we get a completely different protein being produced after that point where those mutations have happened that deleted either 1 or 2 nucleotides in the reading frame. So we call those reading frames because each messenger RNA has 3 possible reading frames. And the reading frame, which one is used, is determined by the relationship and position of that AUG. Because the AUG tells the ribosome, start here, start making the protein from this position. And then the ribosome is forced to read 3 nucleotides as it scans down that messenger RNA. Forced, designed, whatever. So the ribosomes really are little robots. That's what they are in my mind. They click along and they're doing this process, and they're translation machines. And you already know that they're composed of 2 subunits, the 30S and the 50S subunits. You got to do this. Not many teachers are going to force you to go to Wikipedia, but you got to do this. Go back to Wikipedia again. It seriously is going to be the Encyclopedia Britannica of like last year and the future years. And so go to Wikipedia and search for 30S subunit, and you'll be amazed. And then search for the 50S subunit. And if it doesn't come up that way, put 30S ribosomal subunit and you'll find it. What I want you to see there on both of those pages, they have a beautiful model of what those ribosomal subunits look like. And you can see which parts are RNA and which parts are protein. And it's actually 3-dimensional. It's animated. It's spinning around so you can see all the texture and contours of those subunits, and you can see how they relate together. And I think what's going to surprise you is how much of the ribosomes are actually composed of RNA. I want you to look at that so you can figure out how these little robots actually work. And they've got the RNA, if I remember right, is kind of orangey yellowish color, and the protein is a blue color. At least that's what my eyes say. And so go look at those and then imagine them coming together and imagine them doing this process that we're talking about. So the ribosomes have a job. They have to locate punctuation sequences along the messenger RNA. One thing they have to find is the ribosomal binding site. It's a specific sequence. They have to find that. And then they have to find that start codon, that AUG. And once they find that, they move in a 5' to 3' direction along the RNA. And they have to maintain that reading frame, or else they'll get out of line and start making all kinds of crazy proteins. And then basically they bring in the amino acids on the tRNAs, they align them together, and then they form the peptide bond that holds those guys together. And that's the process of translation in a nutshell. So these are tRNAs, and you've got to understand these guys a little bit. And once you look at those structures for ribosomes, you're going to figure out why these tRNAs play a role. The tRNAs come in, and the ribosome, if my iPad is the ribosome is there, the messenger RNA is there, the tRNAs come in, and the RNA in the ribosomes help the tRNAs align in the right orientation so that the rest of the process can happen. Remember, the RNA can base pair, and we get a little bit of base pairing between the tRNA and the ribosomal RNA, and it gets the ribosome and the tRNA lined up just right so that the next enzymatic reactions can occur. So a couple things you need to know, that transfer RNAs or tRNAs, you should know, already deliver amino acids to this area where the ribosome is doing the translation process. You can see this one's charged with a proline, and that's what we call it, a tRNA that has the amino acid on it. We call it a charged tRNA, meaning that the amino acid has been chemically covalently linked to it. And they show it schematically up here. You can kind of see how it 3-dimensionally folds down here in the bottom part of this figure, so you can get an idea of what's going on. So if you think about it this way, each amino acid is going to have 2, 3, 4, sometimes just 1 tRNAs, and the difference between them is going to be predominantly at this position right here, in that part that we call the anticodon. The anticodon has to base pair with the codon that's on the messenger RNA, and if you don't get that base pairing, the ribosome will kick it out and wait for the next tRNA to come in that has the right base pairing to match up with with that messenger RNA. So the RNAs deliver their amino acid, and then they leave it there, and then they can go back and they can be recharged back in the cytoplasm. And there's a whole nother process that is really important to this. If you think about it, if the charging system puts the wrong amino acid on this tRNA, then everything's messed up, right? Because if it puts the wrong one, then the proteins that are going to be produced are just going to be scrambled. They're going to have the wrong amino acid sequence, and one amino acid change can make a defective protein. So that charging system is really important for this process to happen as well. So the tRNAs bring the amino acids in, and we can kind of see that happening. We don't see the tRNAs, but we've got RNA polymerase. They're trying to show you how this works in prokaryotes, because prokaryotes are kind of unique. We've got this promoter sequence on the DNA. RNA polymerase is going to be recruited in there. By now, when we're looking at it, RNA polymerase is way over here. It's already started at the promoter. It starts tracking along. It's actually making this strand of RNA. You can see it right here. And as it's making that strand of RNA, as soon as the ribosomal binding site is produced, ribosomes start jumping on there and start doing the translation process. Okay? And so by now the messenger RNA is only halfway transcribed. But you've already got 5, 6, 7, probably in real life more than that, ribosomes that are already translating it. So you can begin to see how things can happen so quickly in E. coli. As soon as a gene gets kicked on and transcription happens, the first thing that's produced is that ribosomal binding site on the messenger RNA, and then ribosomes start coming and binding to that site and starting the translation process. So when scientists first saw the structure that you see here, and you can imagine in 3D you're going to have polypeptides that are coming off in all different directions, and my lines got a little bit longer, but they saw something that looks like this when they looked under the electron microscope, and they said, hey, that looks a lot like something we use in the laboratory. We use it to clean our test tubes and our bottles, and we call it a bottle brush. And so they called this the bottle brush structure, this structure right here, with those polypeptides being longer at this end where the ribosomes have traveled further along the messenger RNA, and shorter at this end where the ribosomes haven't traveled quite as far along the messenger RNA. As the RNA polymerase continues on down this way, obviously more of the messenger RNA will be produced, and the ribosomes will continue to slide along that messenger RNA until they come to a stop codon, and then that process will complete unless there's another scenario that's set up there. So this is what translation looks like. We have the initiation, and you can see that initiation happening right here, and it all comes back to this AUG that's on the messenger RNA right here. The ribosome, which is depicted by this big blob that's sitting around the message, is lining up with it, and it's waiting for the first tRNA to come in, bringing that methionine in that it's going to bring in. And that's the very first step. One thing I need to tell you that's different between prokaryotes and eukaryotes is the first amino acid that's typically put on proteins in prokaryotes. In eukaryotes, you learned in anatomy and physiology and probably all your other biology classes that methionine is the first amino acid that's put in. But prokaryotes, in the beginning of the protein synthesis, they use a modified methionine. It's called N-formyl methionine. It's got a formyl group on the methionine, so it's a different chemical. And they put that at the beginning of the protein. And that N-formyl methionine is pretty important to you, because when your immune system starts seeing proteins that have a methionine with an N-formyl group on it, and we just abbreviate it FMET because that's easier to say, when they have that FMET amino acid at the beginning of the polypeptide chain, your immune system knows the only kind of cells that does that are bacterial cells. I never use that amino acid. That's an amino acid that's unique to bacteria, and proteins that have that FMET on the beginning of them stimulate the immune response and get the immune response to jump up and get going, because if they're there, if those proteins are there with the N-formyl methionine at the beginning, it means there have to be some bacteria around that are making those proteins. So that's a big signal for your immune system. Then the next thing that happens is that another tRNA comes in, and that's all dictated by what the codon is down here, because the tRNA with the appropriate anticodon will come in. In this case, the codon codes for a proline, and you can see that proline coming in right here. And then the ribosome will slide on down the chain. First, it doesn't do that. It takes the amino acid that was on the first tRNA right here, and it creates a covalent bond between that amino acid and the second amino acid that's being brought in. And then the bond that holds that first amino acid to the original tRNA is broken, and now you've got a dipeptide that's attached to the second tRNA. Now the ribosome moves on down, and another tRNA will come in, and you can see that beginning to happen. The tRNA will come and set into that slot, and then the same thing will happen again. And you can see the ribosome is continuing to move down the messenger RNA, and now we've got a tripeptide. We've got 3 amino acids that have been linked together there, and we've got another amino acid being carried in over here. And as the ribosome allows that to come in and occupy this space, this group over here will be transferred onto that amino acid, that glycine, and then the bond between the first tRNA will be broken, and the process will just keep moving down the line and building that polypeptide in that way. Okay? And then finally, in this case, we come to a codon that is one of those 3 stop codons. Usually, very, very rarely is the ribosome going to be used to make a 4 or 5 peptide molecule. It's just a waste of energy to do that. There are other ways to synthesize that. Usually they're going to be hundreds of amino acids strung together. But now we've got a stop codon right there in that position, and the ribosome kind of sits there and hangs there and says, okay, what do I do? What do I do? No tRNAs can actually bring an amino acid in to put on to the next step. And because that happens, the ribosome cuts the peptide that's been synthesized loose from the tRNA, and the tRNA falls off, the ribosome falls off, and that's the termination of translation in that process. So in eukaryotes and in many prokaryotes as well, following translation, we get some post-translational modifications. Sometimes the polypeptides have things that are put on, like carbohydrates and other things. Other times they just need to be folded a little bit more to be functional by additional proteins that are called chaperones. Some proteins are designed to get pumped outside of the cell. They may be proteins that have to work out in the periplasm, but they're being synthesized in the cytoplasm, and so they have to get stuck outside, and those proteins are going to have what are called signal sequences. They're amino acid sequences, usually at the amino end of the protein, the beginning end of the protein, that say, hey, stick this protein outside the cell. And they're going to be specific amino acid sequences that lead to them being stuck outside of the cell. If they're going to be embedded in the membrane, then they're going to have hydrophobic amino acids that help them hang in the membrane down toward the carboxyl end. And then there are some other sequences that tell them transport these outside of the cell. And many times after they get outside, there are proteases there that actually cleave off parts of those proteins and cut off some of the signal sequences. And those processes are all called post-translational modifications. There's others as well. You learned in AMP about carbohydrates being added to proteins after they were translated. Most bacteria don't do that very much, and so we don't talk about that in this section. But if you're thinking about eukaryotes, adding those carbohydrates to various proteins in eukaryotes like our immune cells, those are very, very important to the function of those proteins. And if the carbohydrates aren't added on correctly, the protein won't be functional in the way it's designed to be functioning. All right.